What is glomerulus? Glomerulus is a very important part of nephron. What is nephron? Nephron is a structural and functional unit of kidneys. There are two types of nephrons mainly, the cortical nephrons and juxtamedullary nephrons. Right? So we have kidneys. How many kidneys we have? Two. And one kidney is composed of about 1.2 million nephrons. It's mean in each kidney there are 1.2 million glomerulus, right? In two kidneys we have 2.4 million nephrons. Approximately this is 2.4 million, or this is from 2 to 2.4 or 5 millions, right? And this kidney is wholly solely dependent upon the functions of nephron. What are the functions of nephrons? Just the functions of kidneys: the filtration of blood, fluid balance in body, absorption and reabsorption of electrolytes. There are some endocrine functions like renin and prostaglandins, erythropoietin secretions, right, and excretion of waste products from body. These are the functions of nephron. How the nephron perform these functions? Nephron have specific parts or specific portions like glomerulus, Bowman's capsule, loop of Henle, the convoluted tubules and collecting ducts. In each of the part there is a specific absorption reabsorption processes occur in nephron, and due to these processes the nephron is responsible for the excretions or all other functions of body, right? So nephron have how many parts? Nephron is composed of two main parts. One is renal corpuscle and second is renal tubules, right? Renal corpuscle is further composed of two parts, the glomerulus and Bowman's capsule. And renal tubules are composed of further mainly proximal convoluted tubules, the loop of Henle, and distal convoluted tubules and collecting ducts, right? So for example, here this is the efferent for example, this lies from glomerulus, right? This is the efferent arteriole and this is the efferent arteriole, right? This is the wall about which we are talking, and this wall is composed of, this is a capillary wall, and this is composed of a very special type of cells which are fenestrated. Fenestrated means there are a lot of holes in the cells of this capillary. For example, these are the cells, right? And here it placed upon a basement membrane, and for example this is a basement glomerular basement membrane, and in center of this glomerular basement membrane we have collagen fibers. Which type of collagen fibers? Collagen type 4, and they form triple helical structures, right? So for example, this is one alpha helical, this is second alpha helical, and this is third alpha helical structures. And here we have the non-collagenous parts of this alpha helical structure, right? So it forms the dense area in the center of glomerular basement membrane, and this dense area is called lamina densa. This lamina densa is composed of triple alpha helix of collagen type 4, right? These alpha helical structures, triple alpha helical structure is composed of type 4 collagen fibers. Type 4 collagen fibers are present in this basement membrane, and I think I have discussed which type of collagen fibers are present in bone. Type 1, which are present in cartilage. Type 2, in basement membranes. Type 4, and other body. Type 3, right? I have discussed this. For example, this is bone, cartilage, and this is the floor. Four under the floor. Type 4 are present under the floor. Under the floor means this is the glomerulus, and this basement membrane is present under this glomerular capillaries. So for example, we can say that 4 under the floor, right? So type 1 is present in bone because the name of bone itself says ONE1. So type 1 collagen fibers are present in bone. If we pronounce this cartilage as cartilage, so type 2 collagen fibers are present in cartilages, and type 4 are present under the floors. Under the floors, which thing is present under the floor? Basement membranes. Like for example, under the glomerular, under the alveolar epithelium, there is also basement membrane, and the basement membrane is composed of type 4 collagen fibers. So 4 under the floor mean basement membrane have type 4 collagen fibers, and all other body have type 3 collagen fibers. So remember this with yourself. This is the lamina densa, which is composed of triple alpha helix of collagen type 4. This is the lamina rara interna, and this lamina rara interna have polyanionic proteoglycans, which make it very negative, right? And this is called lamina rara externa, and it also have polyanionic proteoglycans and make it very negative. Heparin sulfate is also present here. Heparin sulfate, and heparin sulfate makes this membrane very negative. Heparin sulfates are also present on these cells and make it very negative. For example, I am taking one cell from here, this epithelium, right? And for example, this is this epithelium. Epithelium from glomerular capillaries tuft. Why it is very special? Because it have thousands of pores, fenestrations for filtration. So this epithelium is called fenestrated epithelium, right? And this diameter is about 70 to 100 nanometer, right? So these fenestrations are present in this epithelium. And what is the purpose of these fenestrations are whole? Filtration of the wanted substances which is in between 70 to 100 nanometer in diameter, right? And here we have a special type of this is called heparin sulfate, and heparin sulfate have negative charge, so it making this capillary wall very negative, right? And polyanionic proteoglycans making this basement membrane very negative. This is due to polyanionic proteoglycans, right? Now here this triple alpha helical structure is very important. These bulb like structures here, these are the non-collagenous parts of globular proteins, a globular non-collagenous component of glomerular basement membrane, right? And this is very important, right? Why it is important? Because it has some pathological roles. When the body immune system makes some antibodies against this non-collagen parts of glomerular basement membrane, this alpha helical structure will lose its rigidity, right? And this condition is called anti-GBM, antibody glomerular nephritis, right? We will discuss this in last. But here this basement membrane is placed on a Bowman's capsule. So we will take this part of this Bowman's capsule. For example, this is the podocytes of basement membrane. And as I have told you that there are some special types of cells which are called mesangial cells. So for example, these are mesangial cells. These are mesangial cells. And what is the role of mesangial cells? Mesangial cells secrete some connective, it forms some connective tissues. And what is the role of these connective tissues? It gives strength to this glomerulus, right? So first, this is called mesangium. This whole structure is called mesangium, and one cell is called mesangial cell. First, it has some contractility. These are contractile, and it can easily squeeze the glomerulus for full filtration, right? It also produces connective tissues, right? And it also maintain this tuft in place, right? It have many connection from nervous system, like for example, it have a connection from Folgesen cells, and it detects the sodium concentration and will secretes the renin and stimulate the secretions of renin, which is very important for blood pressure maintenance in body. So this is mesangium, and this is the visceral layer of Bowman's capsule, and this is the parietal. For example, this is the parietal layer of Bowman's capsule, and here we have the proximal convoluted tubules, right? This one podocyte and other podocyte, it is connected by a special type of protein, and these proteins are called nephrin proteins. It's called nephrins proteins, right? And here there is some slits between. For example, this is one podocyte and this is another podocyte, right? Here there are some filtration slits are making. For example, this is one podocyte and this is another, and there is forming some slits in between these foot-like processes, and this slit is called filtration slit. Here it is called filtrating slit or filtration slit, and whole this structure is called filtrating diaphragms or filtration diaphragms. These slit have approximately a diameter of 20 to 30 nanometer, right? So this is the, and this cell is called podocyte. This is the visceral layer of Bowman's capsule, and this is the parietal layer of Bowman's capsule. Now, how many blood is entering into this glomerulus or in kidney? 1200 ml. It is about 20 or 22% of full cardiac output per minute, right? For example, we have RBCs in blood. We have RBCs. Also, we have WBCs, and we have also platelets, right? Also in plasma we have albumins. This is albumin. For example, we have globulins, right? And we have fibrinogen, right? In blood we have also calcium, we have also sodium, magnesium, chloride, bicarbonate, iron, and much more, right? Also in blood we have glucose, we have small proteins mean amino acid. Also we have lipids and fats, and we have minerals. Also we have vitamins, right? All these nutrients, electrolytes, and of course we have a lot of water also, right? These all things are present in blood. Also this blood have, the cells have DNA. DNA is have which charge? Negative. It is negatively charged, right? Now this is RBC. What is the diameter of RBC? Approximately the size of RBC is 7 to 8 micron. WBC is 14 to 17 microns, and this platelet is about 2 to 4 or 3 microns. Albumin is about 70 to 100 nanometer, and globular protein fibrinogens are very large in molecular size. These are charged molecule, but they have less charge. Water is polar substance, and glucose, small proteins, lipids, vitamins, and minerals, they can pass from glomerulus. And we have very important thing here which is called creatinine. It is called creatinine. Right? Now, these are types of cells which are present in blood. They cannot pass through the 70 to 100 nanometer diameter of penetrated epithelium, because they have very large size, which is about 7 to 8 microns. Micron and nanometer have a lot of difference. This albumin have very specific and very equal diameter to penetrated epithelium, and globular and fibrinogen are very large molecules, so they cannot pass through this glomerular filtrating unit. These calcium electrolytes, mean calcium, sodium, magnesium, chloride, they can pass through glomerulus. Right? Glucose, small proteins, lipids, minerals and vitamins, they can pass through this glomerulus. So here in Bowman's capsule, when the blood is passing, here we have water, we have sodium, we have calcium, we have magnesium, chlorides and bicarbonate ions, etc. All electrolytes. Here we have glucose, amino acids, lipids and vitamins, etc. They all are present now in Bowman's capsule. The creatinine is passing from this filtrating unit. The solely creatinine cannot go into blood. When you have creat level in your blood, it means your filtrating unit is not working, because it cannot filter this creatinine. Right? So creatinine is also present in Bowman's capsule after filtration. Right? For example, 100% of glucose amino acids are filtered from blood. 100% of water, etc. are filtered from blood, and they are present in Bowman's capsule. Right? Now we will discuss, remember one thing, that glucose, amino acid, lipids and vitamins will reabsorb from proximal convoluted tubules back into blood, right? Because it is needed in blood. For example, this is the efferent arteriole. Efferent arteriole is present here. This glucose, amino acids, etc., they will reabsorb from the urine, and if you have urine or protein in your urine, it means you have dysfunction glomerulus, or your glomerulus or kidney have some type of problems. Right? So here we will divide that reabsorption or absorption of water, electrolytes and proximal convoluted tubules, and loop of Henle and distal convoluted tubules and collecting ducts. They are, in some areas they are hormones dependent, and some areas they are channel dependent, and some areas they are receptor dependent, and some areas the electrolytes and water will travel in between the cells. Right? We will discuss this in detail in urine formation. So this is the first step. This is called ultrafiltration. Why it is called ultrafiltration? Because all the ultra molecules, the macromolecules, the electrolytes, the water, the amino acid, lipids, vitamins, the creatinine, they all are filtered from this glomerular basement membrane, this epithelial layer, also from this visceral epithelium of Bowman's capsule, which are formed of podocytes. Right? Now, what is the role of GFR? What is GFR? Glomerular filtration rate. For example, I have told you that here, for example, this is glomerulus. right? Why GFR is 125 ml per minute? 1200 ml blood is entering into kidneys, 1200 ml. Out of 1200 ml, 625 ml, this is about 20 to 22% of 1200 ml, right? This is enters into glomerulus, and from 625 only 22% can filter from here, which is about 125 ml. So glomerular filtration rate 575 ml goes into efferent arteriole. So 125 ml is filtering from glomerulus. So we can say that normal glomerular filtration rate is 125 ml per minute. This is from here, because one glomerulus is have the ability to filter 125 ml per minute. So GFR is glomerular filtration rate is 125 ml. But this 125 ml per minute glomerular filtration rate is dependent upon hydrostatic pressures here, right? For example, from here, this is glomerulus, right? And 1200 ml blood is entering into this glomerulus. Now the net filtration pressure is dependent upon the pressures which are trying to filter the things from capillaries. The pressure which are trying to filter things from here, and also it is dependent upon the pressures which prevent this filtration, right? So for example, we have net filtration pressure here. We have glomerular filtration rate. This glomerular filtration rate is dependent upon two pressures. The pressures which force filtration and the pressures which prevent filtration, right? First, here is water is present in plasma, right? And it exerts pressures on the filtrating the filtrates to filter it easily from this fenestrated capillaries, and that is called the glomerular hydrostatic pressure. So glomerular hydrostatic pressure. Glomerular hydrostatic pressure is trying to filter things from this and this pressure is about 55 mm of Hg, 55 or 60 mm of Hg, right? Here in blood we have proteins, and proteins are preventing this filtration because protein have very large molecular size, right? So here for example we have albumins, we have the very special is albumin. Albumin have a very important role in this glomerular filtration because it have albumin have 70 to 100 nanometer of diameter, right, which is just equal to the diameter of the fenestrated capillaries, right? So here we have proteins, and proteins are preventing this filtration. It is preventing this filtration, and this pressure which is exerted by the proteins in the glomerular capillaries, it is called colloidal osmotic pressure. It is called colloidal osmotic pressure, which is about 30 mm of Hg. It is about 30 mm of Hg, right? Now this colloidal osmotic pressure is present also imaginary, it is present here, but here it is 0. Why? Anyone? Colloidal osmotic pressure is 0 in here. For example, some pressures are exerting, which is called colloidal osmotic pressures, and colloidal osmotic pressures are exerted by proteins, and in Bowman's capsule we have no proteins because proteins cannot filter from here. So here we have no protein. This pressure is 0 mm of Hg. Now when this filtration occurs, here we have a funnel like structure, and this filtrate is accumulating in this area. For example, it is filled from fluids. When filtration occurs, it will filter from fluids and it will exert pressure from this side to prevent this filtration. And this filtration this pressure is called colloidal capsular hydrostatic pressure. This is glomerular hydrostatic pressure. This is because of water, so it is called hydrated, and this also because of water, and this is in capsule, Bowman's capsule, so it is called capsular hydrostatic pressure, and it is about 15 mm of Hg, right? So glomerular hydrostatic pressure is 55 mm of Hg. Colloidal osmotic pressure in capillaries or glomerulus is 30 mm of Hg, and colloidal osmotic pressure in Bowman's capsule is 0 mm of Hg, and capsular hydrostatic pressure is 15 mm of Hg. Now glomerular, this pressure which force a filtration, we have net filtration pressure. This is the net filtration pressure. This net filtration pressure is equal to the glomerular hydrostatic pressure plus capsular hydrostatic pressure, because these are the two pressures which are forcing the filtration into Bowman's capsule, minus the pressures which prevent the filtration into Bowman's capsule, which are colloidal osmotic pressure plus capsular hydrostatic pressure, right? So put the values. Glomerular hydrostatic pressure is 50 mm of Hg. In Bowman's capsule, this colloidal osmotic pressure is 0, minus colloidal osmotic pressure is 30 mm of Hg, and this capsular hydrostatic pressure is net filtration pressure is equal to glomerular hydrostatic pressure plus colloidal osmotic pressure in Bowman's capsule minus colloidal osmotic pressure in glomerulus plus capsular hydrostatic pressure, which is 15 mm of Hg, right? So this is 50, this is 50 minus 30 plus 15, 45. So the net filtration pressure is 10 mm of Hg. This glomerular filtration occur under 10 mm of Hg, the net filtration pressure. What is the relation between the glomerular filtration rate and net filtration pressure? It is a direct relation, right? For example, this is GFR. GFR is directly proportional to net filtration pressure. If net filtration pressure increases, the glomerular filtration rate will be increases, and when net filtration pressure is decreases, the glomerular filtration rate will be decreases. Now if someone have stone in tubules, If someone have stones, for example, here is a stone, right? Listen it carefully. These fluids will come and accumulate in these tubules, and at last it will accumulate in Bowman's capsule, and the capsular hydrostatic pressure will be increases. For example, this net hydrostatic, this pressure is increases into 20 mm of Hg. Now the net filtration rate will be decreases from 10 to 5 mm of Hg. So when the net filtration rate will decreases to 5 mm of Hg, the glomerular filtration rate will be decreases, right? And that's why by glomerular filtration rate we can examine or we can diagnose the problems in nephrons or in kidneys, right? The diseases which are related to this glomerulus, the first is anti-GBM antibodies glomerular nephritis. Some diseases related to glomerulus. First is anti-GBM nephritis. This is due to when antibodies are produced against these non-collagenous globular components of GBM, glomerular basement membrane. Antibodies are produced. What are the symptoms? The first is proteinuria and hematuria and oligouria, right? Proteins in urine, blood in urine, and less amount of urine. The normal amount of the normal output of urine is 500 ml per day, right? So it will be decreases in GBM antibodies glomerular nephritis. This basement membrane is also present in alveoli. For example, this is alveolar basement membrane, and same antibodies are produced against this basement membrane also, and it will cause hemoptysis. What is hemoptysis? The blood in sputum. Blood in sputum. When you are coughing, in sputum blood will present. This is called hemoptysis, and this is because this basement membrane is called alveolar basement membrane, and this condition is called anti-ABM antibodies alveolar disease. And collectively, if you have hematuria, also you have hemoptysis. These two symptoms. This condition is called good pasture syndrome. And good pasture syndrome, we have hematuria, also we have hemoptysis, we have oligouria, we have proteinuria, and also we have severe cough and chest pain, etc. Right? This is a good posture. This is very bad condition, but why they are called good posture syndrome? It is bad posture syndrome, right? But the name is good posture syndrome when the antibodies are produced against alveoli as well as glomerulus basement membrane. This condition is called good posture syndrome. The next is when some genes mutation occur in, for example, this is the basement membrane, and this is the triple alpha helical collagen type 4, and when some genes mutations occur and this coiling is disturbed, this condition is called hereditary nephritis. Hereditary nephritis. One last, when these podocytes are disturbed, or when this negative charge is neutralized due to some reason, any type of reason, in that condition it is called minimal change glomerular nephritis. Minimal change glomerular nephritis. And in minimal change glomerulitis, we have protein in urine, mainly albumin in urine, because albumin is negatively charged and have large in size. So when these podocytes are disturbed, or these podocytes are finished from here, and this connection is disturbed, and also the negative charges from basement membrane, this capillaries wall, and this podocyte is neutralized, this condition is called minimal change glomerulonephritis. And the main symptoms or main condition in minimal change glomerulonephritis is proteinuria, right? So this is the glomerulus, and these are the hydrostatic pressures, which is very important for glomerular filtration rate. And what is glomerular filtration rate? The rate at which the glomerulus is filtering the blood is called glomerular filtration rate, and which is normally 125 ml per minute. When this rate is increases, it shows some diagnosis in nephrons or in kidneys, and when this glomerular filtration rate is decreases, it also some due to some renal issues, right? Any question? After this glomerular filtration, we will focus on proximal convoluted tubules, which are separate about 1 hour lecture, right? How the water, how the electrolytes are absorbed, reabsorbed, how the glucose, proteins and lipids are absorbed to blood again, right? So we will discuss proximal convoluted tubules in next lecture.